Hi learners, welcome to your own channel BioLearners. At the first video of our channel, let's go and talk about a very basic mechanism or a phenomenon about the muscle contraction, which is known as cross bridge cycle. As we know, all the skeletal as well as the smooth muscle are basically made of with thick and thin filaments. In the thin filament, we can find actin. In the thick filament, there is myosin. And the attachment of myosin and actin and the change of the direction of the myosin tail as well as the myosin head is actually causing the contraction and relaxation of different kind of muscles. Basically, as we know, this is the thin filament and this is the thick filament. And at the resting phase, this thick filament and a thin filament, both of them are present dissociated with each other, or they are not attached. When there is high amount of calcium present in the cell, whatever can be the cell, it can be skeletal muscle cell, or it can be smooth muscle, or it can be anyone. When the calcium ion concentration is high, then what happened? The phosphate group, which was actually attached with the myosin, is removed. And as the expenditure of this removal of this phosphate group, this myosin head is actually attached with the actin and form a cross bridge. So in the presence of calcium, at the first step, what happened? A phosphate group is removed and myosin head, all the whole myosin is attached with actin. And actin is nothing but a part of what? The thin filament. At the next step, what happened? At just the next step, the ADP is being removed. Look at that. There is ADP present in this particular picture. You can see the ADP is removed, and as you see, the head portion is in this particular direction. At the next stage, the head direction is being changed just at the opposite direction, and this is actually done with the expenditure of the removal of this ADP. So at the first stage, we can see at the expenditure of the removal of this PI or the inorganic phosphate, the attachment occur, and the next step, at the expenditure of the removal of this ADP, the direction of the myosin head just changed. At just the next level, what happened? This particular myosin as well as actin remain associated. But how long? A muscle cannot be present in their contracted form all the time. So what cause them dissociate? The ATP. The ATP is actually causing their dissociation. So we can see that the phosphate group removal as well as the presence of calcium causing the association as well as the ATP, which is actually attached with the myosin, causing the dissociation. Okay, and at the begin and at the intermediate step, what happened? The directionality of the myosin head just changed, and this direction change is actually known as what? Is actually known as power stroke. Why this is power? The word power is used. The power word is used as there is a removal of ADP. So with the expenditure of energy, the stroke or this change in the direction occur. This is why this is called the power stroke. At the next step, what happened? This myosin head remain attach with the actin or the thin filament. How long? Until or unless an ATP molecule is attached. When an ATP molecule is attached at the free myosin head, just then, due to the attachment of the free ATP molecule, again there is a dissociation of myosin and actin or thin filaments. And then, as you know, in the myosin head there is a ATPase activity or ATP hydrolysis activity. And due to that ATP hydrolysis activity, what happened? Due to that ATP hydrolysis activity, the ATP again get changed into what? ADP. And there is must be PY. There is also a PI or inorganic phosphate group. I forgot to draw that in this particular portion. This will be the same picture as this. Okay. Now what happened? See this. In this case, the direction is in this particular direction. In this case, the direction again changed. So the myosin head get dissociated from for the attachment of ATP, and then what happened? Due to the ATP hydrolysis, the ATP get changed into ADP and PI or inorganic phosphate, and as a result of that, the head portion is also changed. So the change of the myosin head conformation. So from the whole picture or from the whole cross bridge cycle, there are some take home messages. What are the take home messages? As we can see that the calcium causes the myosin actin cross bridge formation. What do you mean cross bridge formation? Nothing but the association of the myosin as well as the actin of thin filaments. And what making them associate? This making them associate is actually calcium in the presence of what? In the presence of ADP. So calcium caused the myosin actin cross bridge in the presence of ADP. And what caused the removal of myosin head from the actin? ATP is causing the removal of the myosin head from the actin. That's it. These are the two things, take home messages. And there are two conformational changes. As you see, this is number one conformational changes. Another one is this from this. There is number two conformational changes. So the first conformational change of the myosin head, which is called the power stroke, occurred due to the ADP removal. As you can see, there is ADP present and ADP is removed, and as a result of that, what happened? That the power stroke occurred, or the change in the directionality of the myosin head occurred. And the second conformational change occurred due to what? The second conformational change, as you see in the writing, the second conformational change occurred due to what? Due to the ATP hydrolysis into ADP. These are the whole thing. Thank you for watching.